All right, so I'd like to start off by thanking the organizers for the opportunity to speak today, and I will keep this on time. I have practiced it. And I would also like to thank Syngap for the generous funding for the past 2 years to explore a very basic question, which is when and where are neurons actually making Syngap protein? So I am very much a basic neuroscientist, and I am very much interested in a very simple question, which is how do neurons control protein production? And this is a really critical aspect for neurons across both development and in processes such as synaptic plasticity and learning. And over the past 30 years, it's become clear that neurons can actually control protein production, not just in when it's happening, but also localizing protein production to the sites where those proteins are needed in a process called local protein synthesis. And we now know that there's around 5,000 proteins that can be made this way to varying degrees. Now, when I started my group at the University of Edinburgh a few years ago, I was sort of really interested in the fact that if you actually look in this locally synthesized pool of proteins that we now know of, about 40% of these safari risk genes are there. And so there seems to be a high representation of them, and my lab has are interested in a number of them, including Syngap1 and Syngap3 in particular. Now, Syngap1 was an interesting one to us because it sort of bucked the trend of what we typically are looking at with these kind of proteins. We've classically viewed local protein synthesis as a way to strengthen or modify a synaptic compartment. And Syngap is an odd one there in this regard, given that one of its big prominent roles is a negative regulator plasticity. And so a postdoc in my lab took on this challenging project to really start to actually try to understand what the function of making a negative regulator plasticity at the synapse would be. Now, to answer these questions, my lab utilizes a number of tools, and I'll sort of introduce it. I kind of figured for a more general audience, so you guys can have a flavor of what kind of assays we do. We are very interested in using, and there is a backstory of why I have Dolly Parton in my slides, which we can't go into today. If anyone has questions, they can ask me after the talk. So we use transcriptomic-based approaches to actually study the RNAs that are these are localized in dendrites in particular. Increasingly, we're using proteomic-based strategies to pull down RNAs and look at the proteins that are bound to them. In this case, pulling down Syngap RNA and looking at proteins that are associated here. We could visualize the localization of where RNAs and newly synthesized proteins are, particularly in dendrites. Live cell approaches to visualize the manifestation of plasticity and its dynamics in real time, and super resolution approaches. So we could actually probe what's happening at the structure of the synapse itself and look at how different proteins are distributed in time. Now, when we started, we actually had to spend quite a bit of time validating a number of resources to probe Syngap. And so what I'm showing you here is kind of the last 2 years, a lot of the validation, and hoping that we can be very transparent in what we have done to help others so they don't have to go through the same validation. So we've been looking at C-terminal isoforms. We've been able so far to really nicely detect alpha-1, alpha-2, and the beta isoform. And so in our hands in cell culture, using methanol fixation has been really the key to this. We also have been able to get a pan antibody that should recognize alpha-1, alpha-2, and the gamma isoform as well. But for a gamma-specific one, we have not had much luck so far. And it works for classical immunofluorescence, but also for super-resolution microscopy. And so we can see for these isoforms, at least in mature neurons, they are very highly enriched at the synapse. Now, my postdoc, when he first started, wanted to start understanding where is Syngap protein production likely going to be important. And so what he started looking at is across developmental stages of rat cultured hippocampal neurons. So we focused on 7 days in vitro up to 21 days in vitro. These are corresponding to periods where the dendrite is sort of starting to mature, form synapses, up to the point where synapses are fully mature. And what we saw is that Syngap protein generally increases. And what I should say here is that at various points we'll talk about the different isoforms for some specific things. Otherwise, we've been a lot of the time focusing on the alpha-2 isoform, which at least in our hands seems to be the most widely abundant of the three that we can detect. Now, what we are then interested in, well, how does this protein abundance change correspond to RNA changes, and especially localized RNA? So what I'm showing you here is RNA fish data. The top here, we're looking at somatic RNA expression, and we benchmark this to PSD95, which is a classically localized RNA that we know quite about and how its abundance changes. We see generally Syngap RNA is increasing its expression up to div14 and then remains rather sort of abundant, at least in the soma. Interestingly, the dendritic pool of RNA remains rather constant. It's there, but it doesn't really change or fluctuate in accordance to what's happening in the soma, which is different than PSD95, where it's sort of increasing with time. Now, with the ability to look at the total pool of protein in the RNA, we were then very interested in looking at the newly synthesized pool. And this utilizes a technique of metabolic labeling, in this case using an artificial amino acid, azohomoalanine, or AHA. And what we can do is a 30-minute pulse of this amino acid and then utilize the proximity ligation assay, so we could then detect newly synthesized Syngap protein. And what we were really shocked about is that Syngap protein synthesis remains constant through our developmental stages, despite the fact that its protein abundance is changing. And what I should say, in all these cases, we have a negative control where we have inhibited protein synthesis to make sure that we deplete the signal that we see. Now, what we can kind of conclude from this is that as Syngap RNA is increasing slightly over time, the protein is dramatically increasing, and the nascent protein is sort of rather constant, this is likely pointing to that the stabilization of Syngap over time is likely driving its accumulation. However, what we are really curious about is why then bother to increase the RNA at the first place if simply stabilization is enough to drive this. And so what we wanted to then understand is if maybe you were increasing your capacity to make Syngap for a particular reason. And what drew our attention was the reported dispersion phenotype enduring synaptic plasticity. And so we were wondering if Syngap protein synthesis would actually drive Syngap back to the synapse. And this might explain why you're increasing its RNA over time. Now, what we then wanted to do is start seeing if we could replicate this phenotype in our hands, and we were able to. We use, compared to previous reported literature, we use a bit stronger of induction of ChemLTP. We can drive its dispersion. We see that it sort of disperses from all synapses. But what we did notice is something a little troubling, which is that we could only really see in about half the cases where we induced ChemLTP dispersion. And it seemed to be this all or none. Either it was going to disperse or wasn't at all. And when we started quantifying the intensity, we could really see that the more expressed Syngap, our GFP Syngap was, the less likely it was to disperse, which you can really nicely see if you rank order the individual neurons by the intensity of Syngap. Now, this kind of should not be terribly surprising. Syngap is a negative regulator of plasticity. If you overexpress it, it's probably adding a barrier to the manifestation of plasticity. And what we then wanted to start understanding is, well, what's happening to the endogenous proteins? So to start answering this, what we started to do first was look at individual Syngap puncta in images just like this, where we have sort of the baseline state through induction of ChemLTP and periods of recovery. And what we could see is that the intensity of these individual Syngap puncta, now this is corresponding to 10,000s or thousands of synapses for the different isoforms and the pan antibody, is we see really no change in the fluorescence intensity, and this would sort of be a benchmark of the amount of Syngap in individual puncta. Now, this would not pick up if the number of synapses with Syngap has changed. So what we wanted to turn to was a little more sensitive assay to look at this. And to do this, we used expansion microscopy, which allows us to break the normal diffraction limit of light and really peer at what's happening at the synapse itself with super resolution. And what this allowed us to do is look at the number of PSDs that were associated with Syngap and the different isoforms of Syngap, but also look at the relative distribution. So what we can actually do is look at where the peak fluorescence signal for PSD95 is and compare that to the peak intensity for Syngap and see if there's any evidence for maybe more subtle rearrangements. And how we do in both cases, this is a nearest neighbor analysis, and again, in an automated way, so we can analyze tens of thousands of synapses. So off the bat, for these three isoforms, we saw no change in the association of PSD95 with Syngap, which would really suggest that Syngap is still there. Additionally, when we looked at the distribution, we see no change in this. Which would suggest that really there's no rearrangement of where Syngap is falling at the endogenous protein level relative to PSD95. And so what this kind of says, at least here in our hands with this, is that we don't see the endogenous protein dispersing away from the PSD. Some data that I'm not showing here because it's really preliminary is that when we isolate synapses, we see that there is a slight reduction. So there might be a change in affinity for PSD95 that's occurring. But as long as the cell's intact, it's not falling off. But going back to this original question is what's happening to Syngap protein synthesis? And what we initially thought is that Syngap protein synthesis would increase. And what we were sort of flummoxed by is that we saw Syngap synthesis decrease. And this really sort of puzzled us because in the initial 30 minutes after induction of chem-LTP, Syngap synthesis decreases and sort of slowly recovers over time. Now, inadvertently, we actually think we stumbled on why this might be. And this again is going back to our overexpression data with GFP Syngap1 alpha 1. What we noticed is that in a number of cases where we induce chem-LTP, we see signs of excitotoxicity. And this is in the form of dendritic blebbing. This is usually a symptom, especially when you've induced a process like this, that they aren't buffering calcium appropriately. And so we saw in about 33% of cases that we saw signs of excitotoxicity with this protocol, where normally if you're just expressing something like GFP, we would express it to be closer to 2 to 5%. Now, while it's such a small subset that we have here, there is a slight trend towards the higher expression of GFP Syngap, the more likely you are to show signs of this excitotoxicity. So what we think we've inadvertently stumbled upon is that in addition to plasticity inhibiting the function of Syngap, it's also inhibiting the synthesis of Syngap protein. And this sort of negative coupling is likely in a sort of to prevent stress and sensitivity to excitotoxicity. I would love to see if anyone has ideas why this would be, because this is still something that's a little puzzling to us, why overexpression or enhanced expression of Syngap might do this. However, this negative coupling too would also serve as sort of a double-edged sword, because it's why are neurons so sensitive to functional haploinsufficiency of Syngap? Well, if you're artificially pushed into a potentiated-like state, you're reducing the capacity for neurons to recover or enhance Syngap protein production due to the fact that they're negatively inhibiting synthesis. And so what we actually think now is that the local production of Syngap protein is likely helping stabilize and keep this protein amount of Syngap in a nice sort of tight range. Now, what we have been really curious about is understanding how this negative translational regulation is working, with the hope is that if we could actually decipher how this mechanistically is working, if we could sort of modify this or intervene in a very subtle way while keeping the broad translational regulation of Syngap intact, allowing sort of increased functional production of Syngap protein without risking negative effects of overexpression. And so what we've been looking at are RNA binding proteins. So these are classes of proteins that will regulate the entire fate of an RNA's life cycle, including such processes as controlling translational regulation. And so we've identified around 78 proteins that should be interacting with Syngap RNA. And these, so most of these have well-defined functions in RNA biology, some sort of geoclass and geofunction terms here. When we do a string analysis, kind of lets us to look at sort of functional nodes that would be formed. with this. So a lot come up here with splicing. That's where a lot of these regulations are known to occur. But we've identified 4 proteins of interest that are known translational regulators that Reese is right now working on, trying to see if we've removed their binding sites from the 3' untranslated region of Syngap, how that affects their translational regulation, and if any of these are seeming to be negative regulators of translation, to see if we can then devise competition-based strategies that hopefully boost Syngap production. So what we've learned from this is that Syngap mRNA and proteins are highly enriched in dendrites across dendritic and synaptic development. Syngap protein stabilization seems to be what's driving its accumulation and stabilization over time. And we so far can't really see evidence for endogenous Syngap dispersing from the postsynaptic density upon induction of LTP. We do see this negative regulation of Syngap protein synthesis during plasticity, which we think is acting as sort of a protective mechanism. But again, like I mentioned, we're still sort of trying to figure out why this would be. And right now, we're trying to understand how we can utilize this information to enhance Syngap production. So with that, I would like to really acknowledge the first postdoc in my lab who took on this project, Reese. Additionally, our funding, and I promise to update this logo for the updated one. And with that, happy to take questions now and to keep on time for lunch. If you have questions, just grab me at lunch as well.
Do we have any questions? Yeah, we're back there.
Hi, Helen Wilsey, UCSF. Great talk. You mentioned that you had done a lot of validation on the reagents, and so can you talk a little bit about whether those isoform, I assume they're antibodies,
Yep.
isoform-specific antibodies, and how you validated that by immunofluorescence?
So the antibody, so the validation, so this company, CST, was doing a lot of validation on that. We confirmed in our hands specificity in like hex cells and things like that, where we had GFP tagged versions of the specific proteins. So of a GFP alpha 1, beta, that kind of stuff, to do that sort of validation.
And they could discriminate between the two isoforms if you overexpress both?
I think I have to look at what it was like for the beta, because I think that's the more recent ones that we've been looking at. But for alpha 1 and alpha 2, it's very specific.